Hi everyone. So let's go ahead and continue on with this section, which is section 3, looking at the innate immune response or innate immunity. So last time we talked about the first line of defense, which was going to be that physical barrier that we have with the skin and the mucous membranes. And now what we're going to be taking a look at is what happens if if any microbe is able to cross that barrier. We are going to have all of these non-specific internal defenses in place to make sure that it doesn't spread or to slow the growth of the pathogen or kill the pathogen. So we're going to be talking about the activities of various cells. So that's one non-specific internal defense is going to be these different cells that we're going to have. Second one we're going to talk about, included in this video, is going to be chemicals that are going to work. And the third one, which will be another video, is going to talk about other physiological processes that are going to do the job as well, which is inflammation and fever. So let's take a look at some cells. We know these cells, right? We know phagocytic cells. We've talked about neutrophils, we've talked about macrophages, but this might be new, this one right here, dendritic cells. I've mentioned it before, but very briefly. So let's take a look and see how these are going to be different. Neutrophils, remember, are going to be the first ones to arrive on the scene during any type of injury, so during the inflammatory response. And they specialize in eating bacteria. They're really good at getting rid of bacteria. Macrophages are going to come in a little bit later, and they are going to kind of clean up anything that the neutrophils haven't eaten, so any leftover bacteria that's going to be there. They're also going to clean up any cellular debris. So remember that macrophages are kind of the cleanup crew here. They stay longer than neutrophils as well. So again, they're all going to be phagocytic. And then you have dendritic cells. Dendritic cells are going to go in and engulf these pathogens, but they go into the lymph and eventually go to a lymph node so they can present this pathogen to a T lymphocyte. So that's a really interesting concept that's going to happen that we'll talk about later on, the adaptive immune response. So if you take a look at how it works, this is a big giant macrophage. Look at that's a little dendritic cell. The macrophage is going to eat that infectious agent, and once it's inside of a vesicle, it becomes a phagosome. There it is. And here's a lysosome that's going to go in and attach to it. So remember, lysosomes are full of these enzymes that are going to help break down whatever is in this vesicle. Once they are going to be once they are going to be connected, once the lysosome fuses with that phagosome, it becomes a phagolysosome. And that is what's going to go in and destroy that infectious agent. So there's going to be these chemicals that are going to be found, or molecules, I guess I should say, that's going to be found inside of that lysosome. And they are reactive oxygen-containing molecules. And what that does is just it produces this respiratory burst that's going to help destroy that microbe. So once it's destroyed, it is going to be spit out the other side of the microbe. I guess that's a better word, right? Exocytose instead of spit out. Phagocytosis is very effective. Most of the microbes that actually enter our body are taken care of by these phagosomes. So it's kind of interesting, or not phagosomes. Remember, phagosomes are the little vesicles once inside. They are taken care of by phagocytes or phagocytic cells. Let's take a look at some other ones, some more cells. We know, right? We know what basophils are. We've talked about them, but this one might be new to you, mast cells. So mast cells and basophils are going to work very similarly in what they are going to secrete. Remember, basophils are going to circulate around in the blood. There's not a whole lot of basophils found in the bloodstream, but they do circulate in the blood. Mast cells I introduced in section 2, and how they are usually found in the connective tissue like skin and the GI tract, mucosa, and different structures like that. But what they do is they are going to release different granules. So they release histamine. Histamine is going to be important for vasodilation and increasing capillary permeability. We'll talk about what that does later on when we talk about inflammation. There's heparin. Heparin is an an anticoagulant. We mentioned that back in the blood chapter. And it also secretes things called eicosanoids. And what that does is just goes in and enhances and increases inflammation. At the same time, these guys can also go in and secrete chemotaxic chemicals. And what they do is chemotaxic chemicals are going to attract other white blood cells to that area. Sometimes they're also called chemokines, but that's another thing that they can secrete. That doesn't talk about there, but that is going to help with that positive chemotaxis that I had talked about. I think, yeah, it was back in the blood chapter as well. So we will get to these guys again later on whenever we talk about inflammation. Let's take a look at our next cells, NK cells. So these should be new. Again, I had mentioned them before, but we haven't talked about what their specific function is. What they do is they induce apoptosis, which is going to be a cell death or cell suicide. These are made inside of the bone marrow right alongside the other lymphocytes because these are actually a type of lymphocyte. They circulate in the bloodstream, but they do hang out in those secondary lymphatic structures along with your T cells and B cells and macrophages. But since they do circulate around, they're able to do something called immune surveillance. And this immune surveillance is where they're just constantly patrolling the body, and they leave the bloodstream and go to other tissues. So they're constantly in and out of the bloodstream and other tissues, and they are looking for any kind of unwanted cell, whether it's a virus-infected cell, a bacteria-infected cell, a tumor cell. They also look for cells of transplanted tissue as well. So let's say it comes across an unwanted or unhealthy cell, like in this picture. What it does is it is going to secrete these different chemicals. They are going to be these cytotoxic chemicals that they are going to secrete, because they are cytotoxic chemicals are just going to be chemicals that are going to hopefully be toxic to the cell and kill it. So the first thing that it secretes are these things called perforins. Perforins are going to be these neat little proteins that go in and make a hole, a pore, inside of this cell, this unwanted cell. And what it does is it makes a hole for these granzymes. The granzymes are these little tiny dots. The granzymes can enter into the pore and they target the nucleus. And what they do there is they go in and change up some genes. So they turn on and off some genes. So it is going to force that cell to undergo apoptosis, which is cell suicide. It doesn't rupture the cell. It is going to, what apoptosis does is it causes this kind of cell shriveling or cell shrinking. Now let's take a look at our last cell that we are also familiar with here, eosinophils. You remember these? These specialize, not only are they phagocytic, we didn't mention them earlier in the phagocytic cells because they work a little bit differently. They phagocytize allergens, so we'll look at them later on when we look at how they are responsible for allergies and asthma. But whenever they release chemicals, these, again, some more chemotoxic, cytotoxic chemicals that they are going to release, they are going to go in and destroy parasitic worms. So obviously parasitic worms are too big for these guys to eat. So that's why they don't phagocytize these. They phagocytize allergens and antibody-antigen complexes and stuff like that. So when they release these cytotoxic chemicals, they can be cytotoxic chemicals like what the phagocytes release from their little lysosome. So they can be these that are reactive oxygen containing molecules. They can release neurotoxins. They can also release substances like the NK cell with a transmembrane pore, but they don't release granzymes. And again, depending on the parasitic worm, depending on what chemical is released, is going to depend on how it destroys it, but it is going to destroy the parasite. So it's really interesting in the fact that these cells that we just mentioned, the dendritic cells, the eosinophils, how they're able to recognize all of these microbes as foreign because of these receptors. So each of the cells are going to have a pattern recognition receptor, or they're called toll-like receptors. And what they do is they're able to bind to specific patterns on microbe surfaces. So remember, this is a very general recognition. They just recognize it as foreign, and then they are going to be able to destroy it that way. We'll talk about how same kind of concept with T cells and B cells, where they have a receptor and they look for a very specific antigen on the microbe surface. But that is going to be how they're able to go in and recognize that these are going to be foreign substances in the body. Let's quickly talk about pus and abscesses. Who doesn't want to talk about pus and abscesses, right? So pus is basically exudate, and we haven't mentioned exudate yet. Exudate is going to be talked about in inflammation and how it's formed, usually right where that kind of injury has occurred. So it's going to have all of these dead leukocytes like neutrophils and macrophages, but also the destroyed pathogens that these leukocytes have already destroyed as well. Remember, I talked about how neutrophils, once they go in and eat the bacteria and break down the bacteria, they actually die kind of like a honeybee. So that's how pus is formed. Also cellular debris. So pus is really a whole bunch of just dead neutrophils and fragments of pathogens, but with other things in there. Usually what happens is the lymphatic system is able to remove pus, and also, of course, if it's on the surface, the skin is able to remove it as well. But if it's not removed, then it can form something called an abscess. Abscess is going to be where that pus is going to be blocked off by some collagen fibers, and that is going to be where you usually have to have some kind of surgical removal for it, or the abscess has to be lanced and drained and cleaned and all of that good stuff, right? So that's it for our, as far as our non-specific internal defenses, that's it for cells. Let's go ahead and take a look at another non-specific internal defense we have, and these are going to be antimicrobial proteins. So antimicrobial proteins are going to be these molecules that function against microbes. So it's usually going to be these proteins that are secreted from different cells. We're going to just talk about two here in this section. We're going to talk specifically about interferons, and we're going to talk specifically about complement. So interferons, if you remember, I talked about those back when we introduced cytokines. These are going to be a class of cytokines that non-specifically impedes viral spread. So they work at killing off or stopping viral infections from happening. So what happens is that, so let's say that this is going to be the infected cell. Number one is the infected cell. So a virus has already gotten in and it has replicated. When that happens, this infected cell is able to produce these things called interferons. So interferons are going to be kind of like a local hormone. So they are secreted from the cell and they go to their neighboring cells and talk to them. What they're saying is, hey, I've been infected by a virus. You need to save yourself. So here's this normal cell getting the signal from its neighbor saying, hey, there's a virus nearby. Let's make sure that we don't die, or I guess I shouldn't say that. Let's make sure that we don't keep it from, or let's make sure that we keep it from spreading. That's what we want to do. So this normal cell can go in and make these enzymes that are going to interfere with viral replication. So what happens is if the virus goes over here to this normal cell and it goes in and dumps its DNA or RNA inside here, it can't replicate. So, and remember, that's the whole point of viruses, is they need to be able to replicate, but the special little enzymes that this guy has made, because its friend had warned him, they're not able to replicate. So it's kind of interesting how that is going to help go in and stop viruses from going in and spreading like that. Don't worry about the different interferons. As you can see, there's an alpha and a beta. There's a gamma over here. Another thing that they do is they can go in and call other defense mechanisms, these other non-specific internal defenses, these cells, to destroy any virus-infected cell. So here's a phagocyte, a macrophage that's coming in and destroying it. An NK cell is going to come in and destroy it by apoptosis. So it's kind of interesting how interferons work. It's kind of like if you were getting robbed and you're in the closet and calling your neighbor saying, hey, I'm being robbed, so you need to make sure that you save yourself and make sure that they don't come and hit you. So your neighbor is going to, you know, maybe set an alarm or call the police or, I don't know, get their gun or something, whatever your neighbor does, right? I don't know what your neighbor would do. So it's a warning system to tell everyone to kind of save themselves from becoming, from that virus replicating. So now let's take a look at this whole complement system. So the complement system is going to also be a group of antimicrobial proteins. There are at least 30 plasma proteins, and what they do is they work along with or complement, that's where it gets its name, by them complementing antibodies, by working along with these antibodies. Complement, it's the way that it's formed and works, it's very similar to hemostasis in the fact that these complement proteins are made in the liver, just like our clotting factors. They're released in an inactive form, just like our clotting factors. They have to be activated by an enzyme cascade, just like the clotting factors. And that's where it stops because these are important in the immune system. But there's also a couple of different pathways as well. So you don't have to know the different pathways. The book talks about, you know, these couple of pathways, this classic and alternative pathway, and really all the pathway is just their means of activation. You know, one's going to bind to an antibody and the other one's going to bind directly to the microbe, but you don't have to worry about that. What you really need to worry about is just what they do to help defend us against pathogens. And this specifically, the pathogen that they're really good at is going to be fighting off bacterial infection. Very potent at that. So you have something called opsonization that occurs. They go in and help increase inflammation. And they're also going to cause something called cytolysis by releasing these proteins that form an MAC, this little complex that forms a hole inside of the cell. Don't worry about this one right here. So the next video is actually an animation, and it is going to walk you through and kind of show you how these are going to work, what opsonization is. It's kind of tagging of a pathogen so the macrophage can eat it. It'll go through inflammation and look a little bit at inflammation, but we'll look at inflammation here in a second, but also talk about this neat little cytolysis as well. So make sure you watch that animation, and then once you're done with that animation, we're going to pick up where we are leaving off right here. We're going to be talking about the last of our non-specific internal defenses, which is going to be other physiological processes like inflammation and fever. So until next time.